will summarize and talk with you about the findings that we gleaned from all those stored samples of serum and plasma. And what we were looking for are cellular messengers, cellular functions that were changed or produced or evoked by the drinking of the water in contradistinction to plain tap water. And these blood markers were associated with the stress of exercise. And we wanted to know, was there a trail left of these markers that were significant to the stress issue at hand? And so, having established that the water is a superior antioxidant compared to tap water, that it suppressed the inflammatory response, that it acted as an antioxidant, and that it upregulated the mitochondrial activity, and then also that red cell function and so on was improved with the exercise. This is just a summary of what I presented before. We now embarked upon an analysis of cytokines. Now, cytokines are intracellular messages. They come in a variety of proteins. Some of them are what we call redundant. They have the same function but are structurally slightly different. They can act within the cell. They can also act upon through the serum to a different cell, which would almost make them a hormone. But basically, they regulate such things as inflammation and the immune response specifically. Monokines are produced by mononuclear cells, lymphokines by lymphocytes. Interleukins are made by one cytokine and act by one leukocyte, but they act on different leukocytes. And chemokines, thank you. Chemokines are cytokines that have chemotactic. They're attracted by certain chemicals. So we had the report done, testing of serum and plasma for sports study for cytokines and oxidative stress markers. The following cytokines were found to be reactive in this model. We tested for a total of 12, but the others didn't react at all, and that's probably because the stress was specific for certain cytokines, and other stresses would evoke other stresses, cytokines. So we have tumor necrosis factor alpha, which is a very important immune regulating cytokine. And the monocyte chemoattractant protein, that's a protein that attracts monocytes to come and fight inflammation or chronic infection. The macrophage inflammatory protein M1P, which is another one of the cytokines involved in inflammation, interleukin 1 and interleukin 8. On the graphs to follow, tap water is represented in blue, and the kangen water responds in green. Now look at these three cytokines. They all are reduced in activity in the stressed group that drank the kangen water. And this is statistically significant. What does this mean? What this means that the inflammatory markers that we usually see with inflammation are reduced when the recipient and the patient drinks kangen water. And that's significant. So you get more inflammation out of tap water than you get out of kangen water. So that would corroborate the neutrophil story, right? That would corroborate the recovery from stress more quickly. And then we have cytokines that were again, same one, reduced. Now, we also had markers that showed an increase and then a decrease. And this always drives me a little crazy because I tend to get it mixed up. But these are cytokines. They are increased in response, and they're the cytokines that tell the anti-inflammatory cytokines, the inflammatory cytokines to cool down. So you got these up because they tell the other ones to cool down, which makes total sense. But here comes the biggest deal. Again, statistically significant. And this is superoxide dismutase. The most powerful antioxidant made by the human body itself, not CoQ10 or Qunol or any of these things that you buy in the drugstore, actually made by your liver, by your body's immune system, and the liver is a powerful organ in the immune system. Superoxide dismutase is a horrendously powerful antioxidant. It can take peroxide radicals, which are extremely inflammatory, and make water and carbon dioxide out of it. Water and oxygen out of it. Well, that's insane. I mean, it basically is the biggest fighter of oxidation within the human body. That enzyme is significantly increased in response to kangen water. And here you go. That's huge. Look at this activity of superoxide dismutase. Now this, right away when I saw that, I knew number one, it was a good thing. But this immediately kind of demands that we need to study this more, because think about that. That's, not only do you get an antioxidant effect from the water itself, per se, as a direct antioxidant effect, you also get the water to basically kick butt and bring more antioxidants out of the liver. And that to me is huge. And I think fundamentally, that's where we're at, and the scientific evidence now distinguishes kangen water from ordinary drinking water. The properties of kangen water are supported by original research study. Kangen water is effective in human living cell cultures. Kangen water extends into the cell, upregulates mitochondrial activity and function of the mitochondria. It has a proven benefit in exercise-induced stress recovery in healthy human volunteers, with increased red cell production, decreased neutrophil production, appropriate production of cytokines involved in inflammation, and significant increase in superoxide dismutase. So I think we need to look at this in a little different way. We ought to add a few things to what we think this is all about. I would start to say to my prospects, I would try to say to anybody, I will say to anybody, energy is upregulated in the mitochondria by kangen water. Kangen water is an effective antioxidant in single cells and also in the production of superoxide dismutase by the liver. It is an inflammatory, anti-inflammatory agent with depression of inflammatory cytokines and increased elaboration of anti-inflammatory cytokines. Stress reduction and quicker recovery from exercise-induced stress. It maintains the pH balance, proven in mammalian studies and also by our own studies. And it's not an additive, it's a clean, healthy, good-tasting water, free of sugar, dyes, and chemicals, and no side effects. So what's next? We need to publish this stuff. And we are on the way to doing that, but we need to raise some more money for it. We're in fairness, we're in negotiations with the company to cough up a few more bucks. I mean, in all fairness, it's in their best interest. But the rest of the world needs to know this, and the scientific community uses what we call peer-reviewed journals, that are journals of utmost integrity, that have checked the work, that will certify that the work is properly done. And when we do that, more people will know, and it will become a self-fulfilling prophecy. We will have more studies to be done. Sir.
Would that peer review site be, one of them be PubMed?
No, PubMed is just a search engine.
Search engine?
Correct.
It does search once you put in your...
But PubMed only searches peer-reviewed journals.
Peer-reviewed
So if you
journals.
get something out of PubMed, those are good journals.
Good journals.
Junk journals aren't in there.
Okay.
I mean, you can get this thing published in 2 days if you want, but then you don't have that. It'll take a year for that to come out because of the review process that this all goes through. And then, anyway, and the lag time for most good journals, you need to wait 6 to 9 months to get it into, just because of the volume of stuff they have. Okay. So anyway, we're going to publish it. We'll finish. I will publish another version of the book we have now, and we're going to continue our fundraising. And the reason, of course, is that the cost of research is high, and any one of us is not able to fund that. So we need crowdfunding and the company to cough up bucks. But anyway, I want to thank everybody, Fred, Mr. Higa, everybody who has been so good and helpful, and I'll answer any questions if you can stand to listen to me a little longer. Oh, Jane, did you want to say a few words? Or?